Assalamu alaikum, dear students. This is our second lecture of the cell and its function. We are studying the human physiology. Previous lecture में हमने discuss किया था about what is cell, what is the types of cell, what is the cell theory, the modern cell theory, what is protoplasm, what is the inorganic substances, the composition of protoplasm, what are the characteristic or function of the protoplasm, and then we discussed the cell membrane, the plasma membrane, structure of plasma membrane, the types of protein in the plasma membrane. Now we will discuss the other organelles in the cell. We will also discuss that which organelles they are membrane bounded and which are non-membrane bounded. So what are the objectives of today's lecture? At the end of this lecture, you all would be able to enumerate different parts of the cytoplasm. That these are the parts of the cytoplasm. Describe the structure of cell organelles. Explain the function of cell organelles, and differentiate between the membrane bounded and the non-membrane bounded organelles. So let's go and let's go to discuss the cell structure and its function. So cytoplasm. Cytoplasm is the portion of protoplasm which surround the nucleus and peripherally bound by the cell membrane. So we can say that cytoplasm is present between the cell membrane and the nucleus, or the nuclear membrane which is surrounding the nucleus. So nucleus और cell membrane के दरमियान जो कुछ भी है, that is actually known as the cytoplasm. Now, what are the zones of cytoplasm? So there can be the ectoplasm, there can be the endoplasm. The ectoplasm, which is also known as the cortex, portion of the cytoplasm which is present beneath the cell membrane. It contains mainly microfilaments, and the endoplasm, which is present between the cortex and the nuclear membrane. So we can say ectoplasm is the uh, outside portion of the cytoplasm, and endoplasm is the inner portion of the cytoplasm. Now, what are the composition of the cytoplasm? So, first of all, cytosol. What is cytosol? Uh, this cytosol, which is also known as the matrix of the cytoplasm, this is actually the fluid of the cytoplasm. Yeah, we can. This is the watery substance. This is the fluid contained in the cell cytoplasm. The cytosol is composed mostly of water, and it also contains protein, carbohydrates, lipids, and some other inorganic substances. Functionally, the cytosol is the medium in which many metabolic reactions occur, both the catabolic and anabolic reaction, because this is the space where the different uh, reactions in both the catabolic and the catabolic they occur. These the inclusion are the particles. These are the lifeless accumulation of metabolites like stored food, granules, and the pigments. What are the organelles? These are the intracellular minute organs, and these can be again divided into membrane bounded and non-membrane bounded, which will be discussed in the upcoming slide. So cell organelles, the organelles, they are specialized structures that have characteristic shapes and perform specific function in cellular growth, maintenance, and reproduction. These organelles can be the mitochondria, the Golgi apparatus, the endoplasmic reticulum, both smooth and rough, the ribosome, the lysosome. These all are the cell organelles. So here we can see a detailed structure of the human cell. We can see here this white line in the outside. This is actually the cell membrane. This is the cell membrane, also known as the plasma membrane, which is actually the lipid bilayer in which the protein are embedded, which we have discussed. in the previous lecture. This is the protein, and these protein are actually working mostly for the transport of substance, because some substances which are lipid headers, they can go directly through the lipid. So then they can give this protein, they then they will give a pathway for their safe transport across the cell membrane. So this is the plasma membrane, this is the lipid bilayer, and this is equally the membrane protein. The blue color which we can see here, this is actually the cytoplasm, which is a semi-fluid matrix that contains the nucleus. This is the nucleus and all other organelles. This is the mitochondria. We can see here organelle in which the energy is extracted from food during the oxidative metabolism. Here oxidation of glucose or oxidation of the fatty acid occur, which lead towards the production of carbon dioxide, water, and energy. And that energy is used by the different function in our body. This is the secretory vesicle. This is the lysosome. We will discuss in detail in the upcoming slide. This is another structure of the organelle known as the Golgi complex, which actually is involved in the packaging function, and it also distributes those molecules which are manufactured inside the cell. Similarly, these are the centriole. Mostly they are present in pair, and these different substances they are at the cytoskeleton, which support the organelles and they maintain the shape of the cell. And actually, they also play a very critical role in the mobility of the cell. They can be the microtubule, intermediate filament, and the actin filament. The actin filament is twisted protein fibers that are responsible for the cell movement, while microtubule they are tube of protein molecule present in cytoplasm, centrioles, cilia, and flagella. This is the ribosome on the endoplasmic reticulum, these dots, and that's why this can be called as the rough endoplasmic reticulum. This is the peroxisome. It is a type of vesicle which actually can contain a collection of enzymes, and these enzymes are basically used for different function. For example, a detoxification. Any sample of foreign objects, substructures which enter into the cell. So these peroxisome, they just destroy them by their destructive enzymes, by their digestive enzymes. So their detoxification is one of the most uh, important function of the peroxisome. Similarly, the same function is by the lysosome, but lysosome usually also have the digestive enzyme, but it mostly digests the those structure, the organelles which has been damaged or been functionless, so it is being digested by the lysosome, while peroxisome they mostly detoxify the external harmful object. This is the nucleus. In the middle, this is the nucleolus, and then this is the nucleus nuclear envelope, which is being the membrane, nuclear membrane, or the nucleus envelope, which is a double membrane, which is present between the nucleus and the cytoplasm, and that actually separate the nucleus from the cytoplasm. So that was all about some structure. We will discuss them in the upcoming slide. Now, what are the membrane bounded organelles? The membrane bounded organelles, they are the plasma cell membrane, the rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi complex, mitochondria, peroxisomes, lysosomes. These all are the membrane bounded organelles. On the other hand, the non-membrane bounded organelles, they are the ribosome and the filaments, as well as the microtubules, centrioles, and these four organelles they can be called as the non-membrane bounded organelles. Now, what is endoplasmic reticulum? What is its function? We are going to discuss the endoplasmic reticulum. shortly known as the ER. It is a network of membrane that form flattened sac or tubules which are called cisternae. And there are two types, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum, because they contain ribosome on their surface, that's why they are known as the rough endoplasmic reticulum, while smooth endoplasmic reticulum that don't have any ribosome, and that's why they are known as the smooth endoplasmic reticulum. The rough endoplasmic reticulum, they are also known as the granular endoplasmic reticulum, and we have discussed that just because of the ribosome. The main function of the ribosome is the protein synthesis. Uh, so we can see that the protein are synthesized by ribosome on the external surface of the this rough endoplasmic reticulum or the granular endoplasmic reticulum and are then passes to the lumen. Certain cells have more rough endoplasmic reticulum, such as the secretory cell, mucous secretory cell, and swollen secretory cell. They have abundant amount of the rough endoplasmic reticulum. This is the structure of the rough endoplasmic reticulum, which is made of different layers, and these layers are known as the cisternae, and these dots, which we can see here on this surface, these are the ribosomes. What is the function of the this rough endoplasmic reticulum? As the ribosome are present here, and the ribosome main function is the protein synthesis, so we can also consider this function for the rough endoplasmic reticulum. Protein synthesis, rough endoplasmic reticulum contain ribosome and messenger RNA attaches to the ribosome. A triplet of bases is a codon. This is usually the complete process which occur during the protein synthesis. A transfer RNA has an anticodon at one end and the appropriate amino acid at the other. As the code is read, amino acids are assembled into a protein. This is actually the difference between the messenger RNA, the transfer RNA, and then the rRNA, the ribosomal RNA. The rough endoplasmic reticulum, the smooth endoplasmic reticulum, we can see here some if the endoplasmic reticulum have ribosome on their surface, so that's simply called as the rough. If there is no ribosome, they will be called as the smooth endoplasmic reticulum. This is the agranular endoplasmic reticulum. Actually, A means absent or lacking of something. Agranular means the lacking of granular appearance, which means smooth endoplasmic reticulum. Smooth endoplasmic reticulum extends from the rough endoplasmic reticulum to form a network of membrane tubules but does not contain ribosome on its membrane surface. Its main function, as there is no ribosome, so protein synthesis is not a function of this agranular endoplasmic reticulum. So its main function is the lipid biosynthesis and its intracellular transport. In liver cell, a large number of smooth endoplasmic reticulum is found. These play a major role in the glycogen metabolism and detoxification of various compounds like the alcohol, drug, etc. Also found in cells that produce steroid secretion as the renal cortex, the smooth endoplasmic reticulum are the important sites for the steroid synthesis. So from the slide which we can summarize as three main functions: the lipid synthesis, the glycogen metabolism, the detoxification, and the steroid synthesis. Now the Golgi complex. The Golgi complex consists of 3 to 20 stacked flattened membrane sac known as the cisterns. The principal function of the Golgi complex is to process, sort, and deliver protein and lipids to the plasma membrane, lysosome, and secretory vesicle. So its main function is the packaging. It's all involved with the packaging function. This is the Golgi body in a cell, and this is the isolated picture of the Golgi apparatus. These are simply layers, and the structure, the space between these two layers is known as the lumen, and these are actually the secretory vesicles which just come out of the this Golgi apparatus. These are the cisterns. तो ये सिस्टर्नी जो है ये रिलीज करते हैं अ, इस वेजिकल को और फिर इसमें डिफरेंट स्ट्रक्चर्स होते हैं तो इट मीन ये इसमें पैकेजिंग करते हैं और फिर इसको आगे भेज देते हैं देयर आर टू एंड्स वन एंड जहां से ये फिजिकल रिलीज होते हैं तो उसको कहा जाता है द ट्रांस फेस एंड अपोजिट इज नोन एज द सिस फेस सिस फेस भी एक्चुअली जो फिजिकल जो जिसमें स्ट्रक्चर्स होते हैं और इनकमिंग फिजिकल जो है वो आते हैं सिस फेस पे और जो निकलते हैं यहाँ से सिक्रीट होते हैं वो ट्रांस फेस साइड से निकलते हैं so the main function of this Golgi bodies, that is to transport physical bring molecules from the rough endoplasmic reticulum, and when they are transport here, so they will just enter into the Golgi bodies at the cis phase site. Molecules fuse with membrane and are sorted based on their destination. Molecules undergo remodeling and modification in cisternae, and modified molecules are secreted out of cell to another organelle um, at the transphase site. You can see here this is the secretory vesicle and the arrow direction of the arrow indicate that transport vesicle is coming at the cis phase. So its main function is the packaging of the protein and the lipid molecules. इसको गाल्जी बॉडी भी कहा जाता है गाल्जी अपरेटस भी कहा जाता है गाल्जी कॉम्प्लेक्स भी कहा जा सकता है The next is the mitochondria. Mitochondria, they are membrane-bounded cell organelles. इसका singular जो है, this is mitochondria that generate most of the chemical energy needed to the power the cell's biochemical reaction, and that's why it is also known as the powerhouse of the cell. The chemical energy produced by the mitochondria is stored in small molecule called adenosine triphosphate. और जब हमें एनर्जी यूज होती है जरूरत होती है और हमारे सेल्स ट्रांसपोर्ट के लिए मैन्युफैक्चरिंग के लिए ड्यूरिंग द रिस्पायरेशन किसी भी प्रोसेस के लिए अगर हमें एनर्जी चाहिए तो एटीपी जो है दिस इज एक्चुअली और एनर्जी करेंसी ये कन्वर्ट होता है इनटू द एडेनोसिन डाइफॉस्फेट and one phosphate ion is being extracted with the extraction of energy. And then if there is need of more ATPs, so the ADP again it may combine with another inorganic phosphate ion and they will make the adenosine triphosphate. So when three phosphate ion are present with the adenosine ATP and adenosine diphosphate, when two phosphate ion are present with the adenosine known as the ADP and adenosine monophosphate when a single phosphate ion inorganic phosphate ion is combined with the adenosine will be called as the adenosine monophosphate. So this is actually the detailed structure of the mitochondria that just look like a capsule and here we can see here the mid portion inside of these this is the cristae and the cristae actually these are the layer and then inside the cristae there is the structure it is known as the matrix this is the inner membrane space there are two membrane double membrane so this orange color this is the outer membrane and this one yellow one this is the inner membrane and inside the matrix there is DNA as well as some sort of granules and ribosome inside the matrix while there are specific enzymes uh, which which are involved in the production in the synthesis of the adenosine triphosphate. These enzymes are known as the ATP synthase. So these are the ATP synthase particles. Lysosomes, a membrane enclosed vesicle that contain powerful digestive enzymes. Internal pH they are reached up to the 5 and its main function are to digest foreign substances as well as the autophagy. Autophagy form it means those organelles which are useless, functionless and damaged, senile, तो उनकी जो खत्म हो जाते हैं, उनका लाइफ स्पैन खत्म हो जाता है, बूढ़े हो जाते हैं, डैमेज हो जाते हैं, तो उनको जो है फिर ये कहा जाता है, इनको डिस्ट्रॉय करते हैं और इनको रिसाइकल और ये चीजें इसमें डाइजेस्ट कर देते हैं. So this is actually known as the autophagy, yani which mean the cell eating. Recycle on organelles or those organelles which has been damaged. Autolysis, which simply the lysosomal damage after the death. यानी खुद लाइसोसोम जो है ये खुद भी जो है स्प्लिट हो जाता है, डिस्ट्रॉय हो सकता है और खुद को कर सकता है. दैट इज एक्चुअली नोन एज द ऑटोलाइसिस. That was the end of today's session. Thank you so much to all of you. Have a nice day. Allah Hafiz.